Students, we were discussing about various types of sterilization methods which is usually performed with cell culture. And in the last video, we had discussed about uh, different types of sterilization method, and we had discussed about wet heat sterilization. We had discussed an example of autoclave. Followed by that, we had also discussed about dry heat sterilization. Under that, we had discussed about hot air oven, and followed by that, filtering type of sterilization. We had discussed about cellulose acetate as well as PVDF membrane, and the last class we had discussed about membrane filters. Right. So in today's class, we will be discussing about one more type of sterilization, which is with the help of irradiation. So again, irradiations are of multiple types. Basically, the radiation energies can be classified into ionizing radiation and non-ionizing radiation. And ionizing radiations include X-rays, which we all know as an example. And apart from X-rays, we also have gamma rays. And under non-ionizing radiation, we have UV rays and then infrared rays. So in today's class, we will be discussing in detail about how the irradiations methodologies can be used to sterilize the materials that we will use for cell culture. So frequently used type of radiation methodology to sterilize the materials that is used for cell cultures is going to be ultraviolet radiation. So wherein the source for the ultraviolet is going to be a light, UV light basically. So there are three types of rays are there: ultraviolet A, B, and C. And out of this, ultraviolet A rays is going to be mostly filtered by our environment, while B and C rays from sunlight naturally reaches us. And these two rays can also have an effect on the uh, living system. Whereas when we are going to use this ultraviolet rays for sterilizing, that is used for cell culture system, we'll be using a ultraviolet light, which is more or less will be looking like a tube light, but in case of ultraviolet light, it will be emitting UV rays, and it is going to be around 260 nanometer. So this rays, when it comes in contact with the DNA, it forms thymine dimer. So if you have two thymines present in the DNA. So these two thymines are going to be become dimer. So that means there is a bond that is going to be created between thymine and dimer, and hence the microorganism, in case if this thymine dimer happens in the microorganism, the microorganism's DNA nucleic acid cannot be able to undergo further process of preparing the protein. So again, if a microbe has to undergo division, it has to prepare its own protein. Only then it can able to undergo division. But due to this thymine dimer, the microorganisms cannot able to undergo proliferation. So this is the advantage and the mechanism of how ultraviolet rays work. But when we use it for cell culture, the use is going to be not vast. That means it is going to be limited. Why limited? The surfaces on which the ultraviolet rays passes through, only those surfaces are going to be converted into a sterile environment. So in that case, only limited rays is going to be passed through the cabinet, and the cabinet becomes sterilized. But there is a disadvantage part about ultraviolet. One, if we are exposed to it, we will also be able to have a defect in the skin. So you tend to develop a skin cancer, and that is why when there is an ultraviolet rays or ultraviolet light is turned on in a room, you are not supposed to enter the room. And the second disadvantage is it has a poor penetrating ability. That means if you place an object between the ultraviolet rays source and the material of the material that you are going to sterilize, the ultraviolet rays cannot penetrate. So in that way, it has a disadvantage part of using it for a cell culture system. Right. So this ultraviolet rays, as I told you, is going to be frequently used in a cabinet, okay, where we are going to work with the cell cultures. And followed by the ultraviolet rays, we also have something called as gamma rays, and these are highly penetrating. Unlike ultraviolet rays, these rays cannot be interfered by the object. So that means it can able to penetrate through the object, and then it can be able to sterilize the consumables. So in most of the cases, in the industry setup or in a company setup, this gamma rays is going to be used, and frequently bought consumables like tissue culture flask, piper, and so on are going to be treated with the gamma rays. Why? Because even though these consumables are protected by cover, the gamma rays can penetrate the cover, and then it can able to sterilize the material. So in most of the cases, in a normal regular laboratory setup, cell culture laboratory setup, we won't be using gamma rays, we'll be using only ultraviolet rays, while the gamma rays are going to be frequently used in a company setup where they are producing enormous amount of tissue culture consumables. And the last method of sterilization is going to be with the help of chemical, and we call it as chemical sterilization. So we will be using different types of chemicals. One of the chemical which has been frequently used is going to be formaldehyde or ethylene oxide. So formaldehyde is frequently used for fumigation purpose. So in case during the previous classes we have discussed how to sterilize the consumables and the tissue culture wares that we are going to use for cell culture. But what about the tissue culture facility room? The room nook and corner, there are microbes which are present in the environment, which is present along with the gases that are present inside the cell culture room. So in order to uh, remove those microorganisms from the cell culture room, we will be doing a process called as fumigation. So fumigation is a process of creating a gas, and this gas is going to be from formaldehyde or ethylene oxide. And this gas, advantage about this gas is this gas can able to penetrate the entire nook and corners of the room. And this particular formaldehyde will work by uh, interacting, sorry, forming a between two amino acid, it forms cross-linking. So thereby, when the proteins are formed, when if the amino acids are going to be cross-linked with one another, then the protein cannot be functional. So in that case, the microorganisms cannot able to undergo proliferation. In the case of ultraviolet, thymine dimer is going to be formed. In the case of formaldehyde, cross-linking of amino acid is going to take place. So thereby, the protein will become non-functional. Right. So this is the chemical that is used frequently for fumigating the entire room, the entire cell culture room. But one disadvantage about this formaldehyde or ethylene gas is uh, as the temperature increases, as the humidity increases, the efficiency of the gas will also increase. So that is the advantage part of it. That means you have to keep the room at a higher temperature, and you should be having a high humidity. If there is no humidity, then this formaldehyde gas cannot penetrate through the entire part of the room and cannot able to create a sterile room, right? And as I told you, it is usually created in a gas type. Okay, so formaldehyde gas will be produced, and this gas will be circulating entirely through the cell culture room, and will be living 12 hours or 18 hours incubation time. Until then, the gas will circulate through the entire room, and then it will be able to kill the microorganisms which is present in the environment. So this is one of the chemical that we'll be using. And apart from that, we also use hypochlorite. It's also widely used as disinfectant. So again, it's a chemical. So if you are going to use it as a disinfectant, we have to use it on the surfaces where we are going to work. Okay, and a disadvantage part about it is not effective in case if there is a presence of organic matter. And apart from that, it may also able to tend to corrode the surface. Okay, even if it is made up of stainless steel, it can able to corrode the surface. But apart from this disadvantage, it will work efficiently against almost most of the microorganisms which can be killed. And another chemical is going to be phenol. Okay, so phenolic disinfectant, and very popular known company called as Hycolin is already known for us. Most of the cell culture uh, uh, facility will use this high chlorine. It's a kind of a spray which can be sprayed onto the surface, and then we can able to clean it. But the disadvantage part about it is uh, it may leave sticky residues on the surface. Advantage part is, unlike the uh, hypochlorite, it is not going to be lose its activity by presence of organic matter. So in spite of having an organic matter, this particular type of chemical will work as a sterilizing agent. And the last one, which is also being frequently used, is going to be alcohol. So ethanol, propanol is commonly used as disinfectant. And the disadvantage part is it is supposed to be used only to the surfaces. Okay, and we cannot use it for sterilizing the consumables which is present with the liquid media or liquid content. Okay, so surface sterilization only is possible. The effective optimum concentration that has been standardized is going to be 70 to 80 percentage of the ethanol, but usually 70% ethanol is frequently used. So this ethanol is flammable. So when we are working with a Bunsen flame, we are supposed to be highly cautious about using the ethanol alcohol, because it may also tend to undergo burning. So that is the caution that we are supposed to take. But apart from that, we can spray it onto the surfaces, and the entire surface which is in contact with the alcohol will become neutralized, and it is not very effective against fungi and certain bacterial endospores. So in that case, what we are supposed to do, we are supposed to use this UV, followed by that we are supposed to use this ethanol. So these are the different types of methods through which the sterilization uh, can be achieved, and followed by this, we will be discussing about a topic which will be under cell culture uh, topic, which is going to be cell propagation. Okay, so what do you mean by cell propagation? You take a cell and try to grow it so that the cells will start undergo differentiation and will also undergo proliferation, and then you further culture those cells. So we call that as cell propagation. So we will be doing two types of cell culture. One is called as primary cell culture. Another one is called as the transformed or secondary cell cultures. We will be talking that in detail. But first, we will start to discuss about what is this primary cell culture. Primary cell cultures refers to growing a cell which is isolated from a tissue or an organ. Okay, we call it as primary because these cells comes from the live living organ, and these cell cultures can be isolated, cells can be isolated, and then we can grow it in a tissue culture flask. So, but the disadvantage about the primary cell culture is these cells cannot undergo proliferation for numerous number of times. So maybe 2 times or 3 times or 4 times. Beyond that, cells will start to undergo senescence. So the cell will start to die. So, and that is the disadvantage part about primary cell culture. Advantage part, any research that you conduct on primary cell culture will give you an accurate results about that particular organ or a tissue properties. That is the biggest advantage. And if you are going to use a primary cell culture for research work, you have to get a necessary ethical permission, ethical committee permission to work with the primary cell culture. So we call this as primary cell culture, and it is usually derived from a tissue, living tissue or a living organ. Right. So this is how a cell morphology will look, as you can see here. So we have fibroblast cell, we have epithelial, we have endothelial, and we also have neuronal cells. So if you look at the morphology of fibroblasts, you can tend to see that it is elastic in nature. Okay, so it looks like an elastic rubber band. So with the help of this morphology, we can able to identify the cell types also. And but whereas if you look the epithelial cells, these epithelial cells are going to be present in a polygonal structure, and these structures are going to be unique, and these polygonal structures are going to be remain in every single cell. So that is a typical morphology of the epithelial cells. And if you look at the endothelial cells, you tend to see that it is cuboidal in nature. Okay, so these cells will express the morphology of cuboidal structure. Whereas if you look at the neuronal cells, you tend to see that these cells have thread-like structure which are connecting with one cell to another cell. So that is a typical feature of the neuronal cells. How are we going to isolate the cell and how are you going to further culture it? So we call it as in vitro culture. First, you take the tissue and then you disturb this tissue so that you get cells or tissue, and then from that cell we call it as primary culture because we isolate it from a tissue, a single tissue, and then we will do subculture and then it will become secondary culture, and further you do subculture, then it becomes a cell line. So now once it becomes cell lines, there are uh, uh, if you are going to work with primary cell culture, the cells, when you successfully subculture further, it will undergo senescence. It's a natural way of death. Whereas when the same cell line, primary culture, is converted into an immortalized cell line. So this immortalized cell line is also called as transform cell line. So the difference between a normal primary cell and then a transform cell is the normal primary cells will have tight control over cell proliferation. So cell cycle will be tightly regulated, whereas immortalized cell will have loss of control of cell growth. So because there is a lot of mutation that will take place, and that's a complex topics which will be discussed under cancer biology course, right? And apart from this, the primary cell culture, the advantage about using primary cell culture when you do multiple types of subculturing, you tend to isolate a clonal cell line. That is, a single property type of cells can be achieved using this primary cell culture when you do a subculture. Now we know what is primary cell culture and what is secondary cell culture. So primary cell cultures has a typical characteristic features which are listed here, whereas continuous cell lines is also called as transform cell line as a typical characteristic feature. So one of the important feature is primary cell cultures are derived directly from the tissue or an organ, whereas if it is a secondary cell line or a transform cell line, it is derived from the primary cell culture by doing subculturing. And in the previous slide, I forgot to tell you that when the primary cell is going to be converted into immortalized cell line, these cell lines are going to be converted by creating a mutation, as I told you already. So this mutation can be created by radiation or by treating the cell line with certain chemicals called as carcinogens. So thereby these carcinogens will induce mutation to the cell line, and these cell lines will lose its uh, control over the cell cycle regulation. So thereby we call it as transform cell line. So these transform cell lines are always derived from the primary culture. And these transform cell lines are immortalized, whereas in the case of primary, it is not immortalized, it can undergo senescence. And initially, primary cell cultures will be highly heterogeneous, but when you do the subculture continuously, it will become homogeneous. Whereas transform cell lines are going to be always homogeneous cell population, and the transform cell lines will have a loss of anchorage dependent, loss of contact inhibition, and it will have an infinite time, and it is genetically unstable. Opposite to that is going to be the primary cell line, wherein it is going to have a finite lifespan. That means only for a certain period of time these cell lines can survive. Beyond that, the cell will start to undergo senescence, and it will have an anchorage dependent. All the cells will have a interaction with one to another, and if it also exhibit contact inhibition. That means because these cells are under tight control over cell cycle proliferation, one cell can control the proliferation of another cell by contact inhibition. So these are the typical differences between primary and secondary cell culture. I hope this session would have been understanding sessions for you. In this session, we had discussed about uh, two types of sterilization methodology, and we also discussed about what is primary cell. I hope this will make you understand a part about the animal cell culture techniques. And the rest of the topics we will discuss in the next class. Thank you.